Thomas Alva Edison had
His inventing career spanned some 70 years.
He produced almost 1,100 patented inventions.
He made this a century of possibilities.
My name's Jim Melnick. I work at UMDNJ Robert Wood Johnson Medical School. The patent is on our work on autism. And so autism is in part due to genetic defects. And we identified one gene that contributes to autism called INGRAIL2. I remember reading an old New Yorker article talking about autism, Temple Grandin, and they were just talking about how a part of the brain is often malformed in autism called the cerebellum. So I knew lots of mouse mutants that caused similar anatomical phenotypes to those observed in individuals with autism. So I had this idea, let's go ahead and test these genes that cause similar anatomical abnormalities in mice and humans with autism. And it turns out that across campus at Rutgers in the Department of Genetics, the autism samples, DNA samples were stored. And this was run by Jay Tishfield, who was the chair of genetics at that time. I went over and said, hey Jay, I have this idea, what do you think? And he completely supported me, and he's like, why don't you work with this wonderful scientist, physician, psychiatrist, Linda Brustowitz. She's a great human geneticist. So Linda and I teamed up, and we tested some genes, and we got a great result with INGRAIL2, and I've been working on it ever since. One of the wonderful things about science is it always forces you to learn something new, and then science will take you in directions which you never ever expected. And so certainly when I was a graduate student, I never thought I would be studying autism. And so we initially identified INGRAIL2 as a gene, and there are many other genes that contribute to autism. Then we went searching for the genetic variant or mutation within INGRAIL2 that increased risk. We found that. Then we needed to determine, well, how does that work? When does it work during development? And what cells does it work in during brain development? So we've done that. And now we need to understand, well, we understand, we've identified the genetic variant, we understand how it works, but now what are the cell biological pathways that are perturbed? And by doing that, hopefully we'll identify targets for therapeutic intervention.